Bismillahir Rahmanir Rahim. Assalamu alaikum, everyone. I am really grateful to Dr. Danish Bhatti for the opportunity to speak to you. I would have loved to be in person, but unfortunately, COVID-related restrictions are kind of keeping everybody away. So I congratulate you, and also I hope that you will have a wonderful conference. My name is Naweed Syed, and I am a chief scientist here at the Creative Destruction Lab, whereby I'm also a member of the Cumming School of Medicine here at the University of Calgary. Today, I think I'm going to talk about brain and machine interfacing or brain chip interfacing technologies. That is something that is very close to my heart. It's my passion, and this is what I have been doing for the last 35 years or so. I am dialing in from Calgary, which is located in the foothills of Canadian mountains. It's a beautiful city, and we are only a stone throw away from the mountains. And it is an oil-rich place where we have lots of oil. Unfortunately, the oil is in the tar sands, so it costs us much more to extract the oil. We are only 3 hours away from US border to our south side. And so Calgary is a pretty good place. So I am going to talk about something that is very close to my heart, and that is brain. And my interest in brain or brain sciences really began when I was a undergraduate, and then it built on, and ultimately when I became the department head for cell biology and anatomy, I was handed a brain of a young girl. He was 13 years of age, to look at as to what might have happened to her. This young girl was perfectly healthy. She was okay. And she had a little bit of cold and headache, and family thought it was just a cold. They gave her a Tylenol, but it didn't get any better. Her situation started to get worse. She was brought at the Children's Hospital here, and I was the scientific director also for Alberta Children's Hospital Research Institute. When this girl was brought, she was brought into emergency, but her condition started to deteriorate, and then she was moved to intensive care, where within 48 hours she passed away. And they didn't really know as to what might have gone wrong with this young girl who was perfectly healthy. And this brain was sitting in front of me, and I started to think that, this is just a biological tissue. But at the end of the day, everything that she thought of, her, smile, her thinking about her future, her friends, and getting upset for no reason, started to giggle and laugh for no reason, and then her to imagine, demons under her carpet, in her bedroom, and to also love, to be sensitive, affectionate, compassionate. All of those aspects were really part of her brain's doing that were not obvious to us when we looked at this biological tissue. So they were beyond, observable and measurable. So feelings and thoughts, her beliefs, and Santa Claus, the tooth fairy, all of this was really impossible for me to fathom and to realize as to how brain does. So when we think about brain in terms of its functionality, we think about the mindfulness in certain ways. And when the mindfulness is really noticing the details all around us. And that is, what we call the right side of your brain, which is a solipsist brain, that everything you can see, I can feel it, I can touch, I can hear, I can smell. It is here, it is now, and that is a wonderful part of the word. So noticing all the details around us, being present right here and right now. Again, in this particular situation, in your room, you are all connected to one, each other, and that is really what the right side of the brain does. And thinking before acting or speaking, you need to really think about the words. And before you say anything else, that is also part of mindfulness. And then paying attention to how your body is feeling and responding to any particular sensory modalities around you. and that will what we defined as mindfulness. And this is really hard to capture anatomically. So what is brain health? Now, brain health is also the interpretation of senses and the control of movement. And so the right side information is then projected to the left side, and the left part of our brain puts it on the screen, and then it tries to connect it with the past and also connect it with the future to make some sense of all of the situation. And then mental and emotional processing that allows us to be able to figure out all that's happening is also part of our mental health or brain health. And then maintenance of normal behavior and social cognition. And this is also part of our brain health. And a healthy brain will interpret all of these senses, the mental and emotional processing, and also behavior and social cognition. And if it does it under the norm, we call it really the brain health. Now, all of this is really accomplished by tens of billions of brain cells. These brain cells are interconnected, or we call them neurons, and these neurons are more than there are stars in the Milky Ways and the stars in the Milky Ways. So these neurons are in tens of billions, and these neurons that are, of course, electrically excitable, they are connected with each other through special structures. We call them synapses. So the synapses are the meeting point or the talking point between two brain cells or group of brain cells that interact with each other. Now, because these synapses are the communication hub, this is where most drugs are being targeted for diseases such as epilepsy and even Parkinson's disease. We are looking at Alzheimer's disease and the psychiatric disorders, psychological disorders. All of these, because of the synaptic targeting, these are the ones that get targeted. So these neural circuits are, functionally defined neuronal circuits then generate an output which gives a behavior, for example, simple reflexes to complex motor patterns, learning, and memory. So these neuronal circuits, they work together to be able to generate, the output and the neuronal circuit output that controls behavior. But these neuronal circuits are not hardwired. They are constantly undergoing changes depending upon the environmental stimuli to allow brain to adapt to its ever-changing environment. And this is really something important to remember, because this is what will be the focus. The synaptic plasticity will be the focus, because this is something that we work on and we invoke to be able to regain lost brain function or to reprogram brain under certain psychiatric disorders. So these networks and the neuronal connectivity is in part governed by genes, but then epigenetics plays a very important role, whereby environmental factors will determine as to how a particular brain circuit will be put together or will act in concert to be able to elicit a behavior. So these are really the two of the important things. So we know that from neurons or the brain cell circuits, they are interconnected with each other. So that's the middle frame. And they communicate through special structures we call synapses. And these synapses are really the fundamental functional unit of any brain. And these synapses are constantly added or deleted depending upon the utility of that particular network. So come to think about this, after this conference or lectures, you might have gotten several thousand new synapses in your brain, and these synapses will form the basis for learning and memory that we acquire. So it's important to remember that synaptic plasticity forms the basis of all brain functions that we really see in our lives. But these neurons are also not working alone. They work together, or their activities are influenced by the astrocytes of the glial cells. These glia, which were originally thought to be the glue, it actually turns out that they play even more greater role as a tripartite partner to regulate synaptic plasticity or synaptic efficacy. They also release trophic molecules, which enriches the connections between two brain cells. So it is the fundamental unit of a neuron, is a synapse, and the synapse is also modulated and constantly changed by these glial cells. So give you an example as to how adaptive our brain is. I can show you one example where this individual, this student, he actually doesn't know, that this is a rubber hand. So he cannot see his own hand, which is his real hand. He is asked to focus on the rubber hand. And at the same time, when his rubber hand is stroked, his actual hand is also stroked. And once his mind looks at this situation, he would adopt this rubber hand as if it is his own hand. So watch this.
Practice. Here you can see the fake hand, focusing on it. There's the real hand, not focusing on it, simultaneous stroking. And there are various ways you can test it.
So, one more time, I can show you in case you missed it.
Practice. Here you can see the fake hand, focusing on it. There's the real hand, not focusing on it, simultaneous stroking. And there are various ways you can test it.
So the take-home message here is that your brain can easily be fooled, and it is an adaptive mind, so it can be tricked into doing things, and that really allows, again, the invocation of plasticity. So in my lab, what we do is that we train the students using these virtual, augmented, and mixed reality, approaches, whereby we can trick their mind to be able to see things that actually do not exist. So the idea here is the people who have post-traumatic stress disorder, and they are really stuck or have obsessive compulsive behavior or other traits, that you could reprogram the brain by using these non-invasive technologies that allow you to really invoke plasticity that is part of the brain. So she is right now interacting with really another brain which is existing in the space, and then she's trying to locate all the other galaxies and stars, and she could actually reach out to be able to feel. So if it touches the sun, she would feel the heat as well, and it will activate different parts of the brain, that you could use this. So these technologies that are now being used, and they are completely non-invasive, but they invoke on the plasticity of the brain cells to be able to do things. So what I'm going to really talk about today is that, ask the question, how does brain function? And if we are to find out, we have tens of billions of brain cells, and even from simple reflexes to complex motor patterns, learning and memory, we require large networks of brain cells to work in concert. So if one wants to understand how brain functions, one has to acquire the ability to be able to record from large networks of brain cells at a single cell resolution. That is one of the biggest challenges in the field of neuroscience, that you are tapping into information processing capabilities of neuronal network. MRIs and fMRIs and CT scans, all of these approaches will provide you very kind of a gross indication as to which particular area of the brain is involved, but it never allows you to nail it down to a smaller or individual networks of neurons. So if one wants to understand how brain controls simple reflexes, complex motor patterns, learning and memory, it is really important that we acquire the ability to record from large networks of brain cells. And that is one of the grandeur challenge in the field of neuroscience. The other greatest challenge in the field of neurology or neuroscience is that if our brain is damaged, either due to stroke, trauma, injury, Parkinson's, Alzheimer's disease, because natural replacement of damaged brain tissue doesn't take place, how could you regain that lost brain function? Because there are no neurons or synaptic connections that you could generate to be able to fill that gap that has occurred because of the loss of those brain cells. So this is really one of the greatest challenge in the field, is to be able to regain that lost brain function and in cases of Parkinson's, Alzheimer's, stroke, trauma, and injury. So what we thought was that can we, instead of using any other means, can we couple brain cells to a semiconductor chip whereby the damaged brain tissue can be replaced by a semiconductor chip which talks to the brain cells, and brain cells can talk back to it. We know that the stem cell therapy is not going to work in brain. The issue being that when you produce these neurons, they turn into tumors. Also, these newly produced neurons have to project out to targets that are located at some distance. Unfortunately, those molecules that help these axons, dendrites to reach out are no longer there, so they just stay there. And also it is very difficult to pair young cells with older cells, and so stem cell therapy is also not an option. So the only thing that we believe is feasible is to be able to connect brain cells and regain that lost brain function by using chip technology. Of course, it's easier said than done, but to be able to really understand how brain is put together in the first place, we could think about going back to neurodevelopment. So brain, unfortunately, is the fastest growing organ in human body. There are 30,000 synapses every 2 or 3 seconds, and there are millions of cells that are added on an hourly basis. And the brain is the fastest growing organ. So to be able to figure out how it is put together, it is really not possible in the human brain. And there are certain things that we have learned as the time went by. We know that the genetic and environmental factors, they play a very important role in synaptic assembly of brain cells. We also know that many environmental factors can change our brain circuits all the time throughout life. So any perturbation or any alteration to this synaptic connectivity can render the brain circuits dysfunctional, which could lead to neurodevelopmental disorders such as autism spectrums and etc. So we have learned that a number of model systems, even from primates and in rodents, that neurons or the brain cells that fire together, they wire together. And we learned this example from humans. If you put a patch over a kitty cat's eyes soon after birth for a few days, that kitty cat will be blind in that eye for life. So the sensory input is really important for visual-tactile pathway development. And similarly, children who cannot hear, they cannot speak, because the cells have to fire together in order to wire together. So neurons that fire apart, if you perturb the activity, they will fall apart. They will not be able to connect appropriately, and as a result, you have developmental deficits. Similarly, neurons that fire out of sync, and we see this in the case of, for example, epilepsy, these circuits will then fail to link, or the neurons will fail to link, and that results into diseases such as epilepsy and many others. So when we think about really the long-term impact of environmental factors, a child or 3 years old, this is the brain of 2 brains of 3 years old boys. On the left, which is a normal brain, this child was loved, cared, taken care of, nurtured, provided with an excellent environmental factors for brain to develop normally. The child on the right-hand side underwent through extreme neglect, and this is abuse and neglect, and then also chained and hurt and really not allowed to develop social and emotional and intelligence. The mental health was seriously compromised. That child's brain never really developed, and you can see that the big gaps from the left and right side of the brain, and these do not allow them to be able to really develop as a normal human being, as a normal adult. However, plasticity once again can play an important role if these children are provided a learning environment, and love and compassion and enrichment. This could overcome some of the effects. but there is a narrow window during which we could do this. So this is something to really remember. Similarly, neurons in the case of Alzheimer's disease or Parkinson's disease, when they do not fire in sync, they fail to link, and then the circuit really falls apart. And that results into lack of trophic molecules, lack of activity, which results into tau and alpha and A beta accumulation, fibrillary entanglement. Then the cells will eventually commit suicide and they die because of that. So many aspects of the brain development requires, proper connectivity patterns. Not only that we require connectivity patterns and the activity patterns between like-minded cells to connect with each other, but also their continuous functioning is contingent upon these brain cells continuing to communicate with each other. And I think COVID-19 has taught us that how isolation, especially in our part of the world, has resulted into creating brain fog, which really perturbs the normal capacity, the functionality of the brain. So we started to really think, and during my PhD, I recorded from 12 brain cells, and I thought that, I walked on water, only to realize that I was the dumbest person who could think that recording from 12 brain cells at the intracellular level, I might be able to figure out or crack the code or the holy grail of neuroscience or neuronal connectivity, only to realize that I was far, far away from that. So we started to think about how can we start to record activity from large networks of neurons? And if we can do this, we will be able to really recapitulate that activity and then design semiconductor chip that would be allowed to really act and mute just like brain cells or brain circuits. And in the future, the idea is that then the damaged brain part of the tissue can be replaced by that semiconductor chip. So brain chip and computer interfacing was a crazy idea. When we thought about this, everyone thought that we were just crazy, because the human brain, as I said in my first slide, is biological. To connecting it to any other device, would be a futile and then really a crazy idea to do this. But we went ahead and we said that, we could perhaps use these disruptive technologies. And if we can recapitulate the interworking of the brain, and of course the brain is electrical, it conducts electrical impulses, and it has transistors, capacitors. These are all the capabilities which form the basis of an excitable membrane. If we could recapitulate this, this could be a really cool tool for us to be able to tap into information processing capabilities of the brain and its functionality. And once we know from recording from the intact brain as to how networks of brain cells function, it is really then very easy to be able to recapitulate this network processing using artificial devices such as a semiconductor chip. And we tapped onto the idea that we are natural cyborgs. We connect with devices naturally. So brain plasticity allows us to attach ourselves to machines such as computers and electronics quite naturally. So the first step really was that using a non-invasive approach, can we record brain activity just by, really putting electrodes around the scalp from the outside, not invasive, and do this in humans. So our brain automatically reconfigures itself, as I showed you earlier, in response to an input from simple tools that we use routinely. So, we pick up our phone, we just, connect to our phones. We know how it is connected to server, it's connected to the main hub, and it is connected to the satellites and the tower. And then the whole thing automatically, spontaneously connect. We connect to our devices naturally. And it is far more open system that we ever thought that our brain could be a closed circuit. It is actually not. As I talked about plasticity, and brain also survives in the changing environment itself. It changes itself and it adapts itself to survive in that particular environment. So when you think about brain-machine interfacing, when we think about doing an action, so for example, if I were to reach out to grab my remote control, And if you are monitoring my brain activity from the outside, the same area of my brain, the same brain cells will discharge and fire if I imagine doing that action. So if I imagine that I'm reaching out to grab my remote control, the same part of my brain will be activated. So if I put electrode, I can only imagine doing an action, and that action will really come to life. And if I could use my brain as an interface that is serving as a remote control, and I connect that remote control to a drone or a car, I can actually drive it by just imagining it. And I'll show you some of that shortly. So thought translation machine, they tap into motor program in a person or animal's imagination and act, and decode that distinctive electrical command to a device that put the thought into action. So you thought it was really hard? It's actually not. It's very simple. So the way you could do this, or it works, is, for example, we can use these robotic monkeys that in our lab, whereby what we can do. This is a robot, and Thomas here has got these electrodes around his scalp. So whatever he will do, and imagine, the monkey will just copy it. So if he moves his cheeks or lips or his eyes, or and even he could turn on the light and turn light on or off by just imagining that he's turning the lights on or off. And we can bring in young kids who have never been trained. We put electrodes, and whatever they will do, the actual monkey will just replicate his thoughts, converted into an action into a computer. So we train them to control this cube in a tunnel, which allows him to lift the cube up or down, left or right, and it pushes it deeper into the tunnel, which really reflects the forward motion. So what it really does is that we can think about car to move, and then the car move with exquisite accuracy. And he's controlling all of this car with his own mind. And we can do the same thing, and we can control, for example, a drone by just thinking about it through our mind control. And in a very tight setting within my lab, we can fly these drones around, and somebody is watching the door in case Dean is passing by. We're not supposed to do these things, but we do it anyway. So on these little drones, we put in small sensors, and these sensors are the cameras. They provide constant information to the electrodes around the scalp, and they are both in sync and in communication. And the constant back and forth, the thinker is now thinking, and as a result, you can actually land on this drone by just using your brain power. So the mind controls, many of these objects, and I'm leading you slowly to where we really wanted to take this. So right now we're recording from the scalp outside, but it is not at the level of single neurons still. It is at the level of large populations of neurons that are discharging, and these electrodes are picking up population spikes and doing simple actions, for example, flying a drone. So the idea really is that, can we tap into individual neurons at the single cell level, whereby we have much greater control, and also working at the single cell level allows us to be able to really replace only those cells that have been damaged either due to stroke, trauma, or injury. So these bionic hybrids, for example, to give you an example, in a situation where you have people who have Parkinson's disease that didn't respond to any drugs, the surgeon goes in and implants a deep brain stimulation electrode. The electrode goes deep into substantia nigra. The patient has a button, and whenever they would like to stop their tremor and want to do something, they stimulate the electrode. The electrode stimulates those neurons, and oftentimes, the dopamine release occurs, or the synaptic communication is enhanced, and as a result, those tremors will stop. But in this particular case, it's a one-way communication. Electrode is stimulating the parts of the brain uncontrollably. Sometimes excitotoxicity results and the corrosion of electrode also damages those normal parts of the brain. And the cells are screaming, stop, 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 but there is really no control mechanisms to stop the electrode from excessive or overstimulating. And the other case on the top, you see the right-hand side, when you see these neurons in, for example, scalp electrodes or the depth electrodes, they are only monitoring the activity or the epileptic discharges in patients who are, firing out of syncs or having an epileptic seizure. But there's nothing that they could do. What we wanted to do was to create a true bionic hybrid, whereby, as shown in B here, we could stimulate individual neurons at the single cell level using capacitive stimulation. And when this cell is activated, it is connected to other cells. When this one fires, we have a transistor underneath that picks up a signal. So we complete a loop whereby an electronic device can talk to brain cells, and then brain cells can talk back to those electronic device. And this we thought was absolutely essential for any brain control, prosthetic devices or brain machine interfacing, or to replace the damaged tissue. Because in instances, if these two brain cells had lost their contact because of stroke or injury, we could easily bypass it by using a transistor to a stimulator, which is the capacitor, and to activate these neurons to really fill the gap that has happened because of the damage of the brain tissue. So how does it really work? This is our very old chip, whereby we could, place these neurons, of course, from animals, isolate them from brain, you put them on top of capacitor, and when we stimulate it through the capacitor, brain cell is activated, and as if another cell is exciting it. And if two cells are synaptically paired, we can activate a single brain cell, the impulse will activate this postsynaptic neuron, and the transistor underneath picks up a signal. So you complete a loop whereby an electronic device can talk to brain cells and they can talk back. And that was really, I never thought we will live long enough, or I will live long enough to see this, but that was really major breakthrough. But the question still was that this is on a shorter time scale or basis. We wanted to make long-term recordings so we can monitor these entire network assembly to come together. And as they are coming together, we are constantly monitoring their growth as well as their activity. Now that was really an incredible feat. We designed these incubators that have CO2, PCO2. So basically all the stuff, the goodies that you have in your brain, we recapitulated onto these incubators, and we could grow these brain cells and monitor their networks formation from days to weeks to months. And this provided us some very unique insights about neural network functions, and we could continuously monitor their activity. You can see these populations of these neurons spiking, and whenever they will spike or discharge, we will know which particular electrode is actually communicating with particular groups of neurons. And we could do this for weeks and months, but these are neurons that are in contact with the chip. And you could say that, well, what about an intact brain which Mother Nature has put together? Because when we dissociate these neurons from rats or from mice, two neurons will probably never connect with each other in reality. So we can take the entire intact slice and we could record from intact slice of the brain at the single cell resolution. And this allows us to be able to monitor the activity of large networks of neurons. So that was the second level of difficulty that we achieved, that we could now record from networks of brain cells that are naturally synaptically connected in the brain. But still we weren't happy, because brain is 3-dimensional. We are only recording from these cells that are in immediate contact, but brain is 3-dimensional. So we developed another technique, which we call photoconductive stimulation, whereby we can grow these brain cells on silicon wafer. We label them with the fluorescent dye, for example, a calcium indicator or voltage sensitive dye. Whenever this cell will fire, it will just light up like a Christmas tree. So what we could do is that we fire a laser through the microscope. the actual laser changes the properties of the wafer, and that will depolarize or activate a neuron. And when it is activated, it will really turn into this. So a single pulse will activate these large networks of neurons, and we can follow the activity as it goes from one cell to another through synaptic connectivity. So now we are monitoring large networks of these brain cells at a single cell resolution. This allows us to be able to record from several thousands neurons that are in immediate contact with chip, and also monitor the activities of a larger network that is sitting above the chip. And if you put it all together, really, this is what it comes down to. And this is what is happening now, whereby this particular monkey has actually no arm. Its visual cortex makes contact where the food palate is. The visual cortex talks to sensory motor cortex. And when sensory motor cortex discharges, you have electrodes, so the chip planted inside, and that chip becomes a remote control that can now control this particular artificial or prosthetic limb. Now, these technologies have really grown quite significantly since the original inception of them, whereby now the nanofibers conduct speed at the light of light, and at the same time, the G5 technology allows for this monkey even to play ping pong. So these are really something that happens in instances where people lose their arms or legs. Your brain is still intact. You have no organ attached. So if you could think about planting a chip inside the brain, in the sensory motor cortex, the brain will talk to the chip, and then the chip becomes a remote control, which can then drive that prosthetic limb as if it is part of your own body. So brain-controlled prosthetic devices will soon become a reality. And I think this is where we are heading. But to be able to get into humans, it's not that easy to really take a human brain and start planting chips into. There are regulatory issues are really very stringent. And we also don't want to damage even normal part of the brain to be able to really plant a chip. So we first embarked on seizure detection because skull can be opened up, the electrodes can be planted subcortically, and we can monitor seizure-like activity in the first instance. And that's what we really wanted to start our technology. And we developed these 3D electrodes, and these electrodes are almost several hundred times more sensitive than the surface electrodes that are used now for detection of epilepsy in children. The problem in that particular case is that the child has a 30-foot cable, and child is staying in the hospital to be able to pick every single seizure, or for surgeon to determine where the seizures are coming from. So it is really not very sensitive or not very accurate. Often surgeon ends up taking the normal tissue, and this leaves the functional deficit in a child's brain. And then the child is also stressed in the hospital as well. So what we wanted to do was to actually develop flexible electrodes that are much more sensitive than ECOG electrodes that are commercially available. We also wanted to make them completely wireless so the child can go home. We also wanted to make sure that these electrodes are MR compatible. So once these 3D electrodes, which actually protrude into the tissue, and they provide an excellent resolution. We tested this in rats first, these electrodes, and this paper just came out in one of the Nature Family journals. And we implant these inside, and we then we recorded both spontaneous and induced seizures in these rats for up to 8 months, and with exquisite accuracy. So once that was validated, this technology won several awards. We wanted to move forward to be able to see if we could actually do this in humans. So our team of biomedical engineers, as well as surgeons, we all got together, and we said to the surgeons that as you are planting these surface electrodes to monitor seizures, we will piggyback and we will plant our flexible electrodes at the same time. So you don't have to do any other surgery once you open up the brain, and then we will make it completely wireless. and we will also make it completely MR compatible. So once these electrodes are mounted around the scalp, one could do is that because they are MR compatible, you could do an MRI scan, and so that will tell you exactly where the electrode position is in the context of different parts of the brain. So the idea really is that if a seizure is triggered, the electrodes will pick up the electrical activity, and the MRI image, could then be coupled with to create a tomogram or a 3D rendition of the entire brain area vis-a-vis the context of different electrodes. And what that does is that it provides the surgeon information. For example, these are completely wireless, they are flexible, they are not really rigid, they could be attached to the surface, and this little contact outside acts as a wireless device. The child or the adult will wear a portable device, a backpack or a cell phone, which will pick up every single seizure with exquisite accuracy, and then create, and our machine learning algorithm will create a 3D rendition to provide surgeon an image every time a seizure occurs, and tell the surgeon that out of 30 seizures, 29 of them have come from exactly this place. And this is how you could really design surgery to be able to remove them. So you pull it out of the scalp, and we were able to monitor every single seizure with exquisite accuracy at the resolution of a single, subsets of neurons. So this is really a remarkable thing in this context. It's going through clinical trials. So we are pushing forward with this technology. Now the idea that we are developing is that to use same electrodes, and now instead of getting them just to monitor, they also become the interceptors. So as soon as the electrical discharge or these epileptic seizures spread, we should be able to turn these electrodes from a signal monitoring device to actually suppression of that activity by passing hyperpolarizing or negative current pulses. We are in negotiation with iPhone and Apple, whereby we are designing a cochlear implant which is sensitive enough to pick up seizures even before a patient is aware, because the perturbed or the artificial intelligence devices and also machine learning algorithms allows us to be able to predict abnormal activity before it becomes a full-blown seizure. And then that implant can pick up the signal, dials your cell phone and tells you you're about to have seizure, settle down, and then it turns on, it dials 911 and turns on the GPS locator on your phone to tell you exactly where you are, or the ambulance where exactly you are, and then the intervention can stop. So I think from a detection device, it could also become an interceptor device as well to block. So this is what your future doctor is really going to look like. And I think the way these artificial intelligence and these brain machine interfacing technologies are coming online, I think the world will change completely, not just for the neurological diseases or developmental disorders, but also I think in terms of recreating the brain that has been damaged or people who have died. So I will end by showing you this video, which I think is not that far, maybe 30 years down the line.
It takes a moment for the unthinkable to become reality. In the near future, tragedy may come with new choices. Imagine you've been in a car crash that's killed your daughter.
Is she okay? Is she okay?
Or has it?
She's breathing. She's breathing.
Hey.
Is she okay?
Time of death, 9:17. Your daughter's brain was not impacted by the accident. If you wish to scan her brain for digitization and upload, you have approximately 5 minutes.
Do it.
Now imagine the power you wield when you can order a digital copy of your daughter's brain. And do you know what comes next?
Hi, Dad.
One day delivered to your doorstep is an artificially intelligent android that will act, look, emote, just like your daughter. Every memory from birth on will remain intact, but she'll also be a walking computer with access to all the world's knowledge. This is what your world will look like. This is the future of artificial intelligence.
Thank you. I think now that I've scared everybody, but one thing is for sure that these technologies are still futuristic, but many aspects of these technologies are really coming online. But there are some things for that you always need Shadi.com. So thank you very much for your attention. I'm sorry I've gone a little bit over. It's hard to really speak to a wall or a computer not having the audience or the timer in front of. So I hope that you will enjoy your conference, and I would have loved to be there to answer your questions or alleviate some concerns, or maybe make you a little bit uncomfortable in terms of where the neuroscience or the neurology is heading. So thank you very much. Thanks again for the opportunity. Best wishes and best of luck.